Hello, I'm Marketa from CEITEC, and today we are here with Marketa Suranova from Brno University of Technology. Hello, Marketa.
Hello, Aneta.
Marketa uses QPhase, the holographic microscope, for live cell analysis in her experiments. So, Marketa, what exactly do you study?
So I am studying microstatics, which inhibited and stop migration of cells from tumor. For this research, we used, for example, vincristine, doxycycline, 4-hydroxyacetophenin, and many other drugs. And I used use for these drugs cell line of human lung and human breast. So I interested in biophysical interpretation of quantitative phase imaging using by holographic microscope QPhase.
In your experiments, you are dealing with drug effectiveness. What are the advantages of quantitative phase imaging over other methods?
Thanks to QPhase microscope, I analyze dynamic and morphological changes of cells with drugs or without drugs. And the advantages could be that we use free objective lens. For dynamic analysis, we use objective lens 10 times because we have a very large field of view. And next we can use objective 20 times and 40 times for check the morphological changes in the cells. Because we can see the vacuoles, membrane, nucleus, and many other parts of cells.
So, do you use any fluorescence labels, or do you see these organelles without any label?
Yes, because this microscope is multimodal, so we can use for better analysis fluorescent mode.
Okay, and what research difficulties do you encounter?
Difficult could be that we have a very big data set, because very often I use this microscope for very long time, for very long measurements.
Okay, how long experiments do you do?
20 or 24 hours in 5 minutes interval. So the data set is very big. But on the other hand, we can use software QPhase for analysis, and there isn't very big problem.
Okay, are you satisfied with the user interface that our software SophiQ offers?
Yes, I use quantitative phase image for the analysis in this software.
Okay, the software offers many parameters for cell analysis. Which one do you use most often?
For dynamic, I very often use speed, growth speed, elongation, spreading, perimeters, and mass, RL, and many other parameters. And very interesting for us is motility rose graph, because we can compare the moving of control sample and sample with drugs.
That's interesting. Do you see QPhase as an intuitive device, or how long have you been training?
I have been training maybe 1 month.
Okay, it's not so long. So, what stage are your research at now, and what do you plan to focus on in the future?
Now we have lots of data sets. We are doing analysis, and we prepare the publications. And in the future, we would like to continue with microstatics, and we would like to test collagen, for example, because the very advantage of this microscope is that we can see the cells in scattering medium.
That's true. So, thank you very much for your visit.
Thank you, Aneta.
We wish you good luck for CEITEC. And the audience may be interested that they can try our microscopes, CEITEC QPhase, in laboratories of Masaryk University or CEITEC in Brno, and also in Institute of Molecular Genetics in Prague.